So let's move on to mitochondria and chloroplasts, also known as energy-related organelles. And these are considered energy-related organelles because both of these organelles allows us to take an outsourced energy. So in eukaryotic cell, we can consume our energy, and by consuming energy such as glucose and fructose, we're able to then convert that glucose into ATP within the mitochondria. In plant cells, as well as certain proteins, also known as chloroplast proteins, and algae, they also can have mitochondria, but additionally, they have chloroplasts. So chloroplast right here is this organelle that allows us to take sun energy and convert that sun energy into glucose, and then that glucose can later on be converted into ATP for these organisms to then use as an energy source. So before we dive further into mitochondria and chloroplasts, let's take a step back and look at the endosymbiotic theory. So the endosymbiotic theory is a theory that suggests, that hypothesized that organelles originally existed as these free living prokaryotes. So we learned before prokaryotes includes bacteria and archaea. So these free living prokaryotes were living, and the theory suggests that these prokaryotes were then engulfed by larger prokaryotes, larger cells, and in the process of being engulfed, they became organelles. So what we can see here is that you have a possible aerobic bacterium. So this would be what later becomes a mitochondria. So I'm going to write M for mitochondria, right? Or we can have the cyanobacterium. The cyanobacterium have an ability to use photosynthesis to convert that sun energy into chemical energy. And so the cyanobacterium, when they're engulfed by this larger cell here, later then becomes the chloroplast, right? So this is our ancestral host cell, which was a larger cell that then engulfed these smaller prokaryotes, and these smaller prokaryotes, both of them here, benefit because this larger cell provide for it protection. It also provides a bunch of nutrients within this cytoplasm here, and these chloroplasts and mitochondria also provide a benefit to the large ancestral host cells, because the large ancestral host cells now can get ATP that are derived within the mitochondria and chloroplasts. Okay, so this would be an example of monocell. And so real quick, so this video right here, we'll talk a little bit more about endosymbiotic theorem. It's actually a really cute video, so I hope you guys take the time to take a look at it. But what I wanted to say is we call this an endosymbiotic theory, and I wanted to touch upon this one real quick. So just keep in mind that theory, a hypothesis is an educated guess, right? But a theory is not an educated guess. At this point, a theory is explained by different scientific events. So we make an observation, but then we're able to explain that observation using many different evidence to support our hypothesis. So once it becomes a theory, it has been tested and proven many times, been supported many times. And while a theory can be altered at a later time as we get more evidence, there is enough testable evidence that suggests that what we observe becomes a theory. What we observe to be true becomes a theory, because a theory is those scientific experiments have supported enough time our hypothesis to actually show that we now consider it as a scientific explanation of what is observed. Okay, so let's move on to a little more detail about mitochondria and as well as chloroplasts. So let's start with mitochondria first. So mitochondria is this potato-looking thing. I think it looks like a potato. This potato-looking thing that has a double membrane. So similar to a cell, we have a double membrane there. And this double membrane system allows us to actually carry out very efficient energy releasing reaction. So these reactions, basically reactions that are needed in order to convert that glucose into ATP. Keep in mind that these reactions are aerobic. So here we're doing aerobic respiration. And aerobic respiration means that it requires oxygen to complete the reaction. If it is not, if oxygen is not present, we can still make energy, but it's not as efficient as the energy that we make in aerobic respiration. So those are known as anaerobic respiration. And there's two main type of anaerobic respiration. So in eukaryotic cells, we do lactic acid fermentation. So this doesn't require oxygen and is less energy efficient compared to aerobic respiration, but it's able to convert that glucose into pyruvate and move that pyruvate toward a lactic acid fermentation. In yeast, we do what's called ethanol fermentation. So again, this doesn't require oxygen. And as we saw when we were looking at macromolecules, we see that yeast can actually take maltose, convert that maltose into 2-glucose, and then take that glucose and convert it into ethanol and carbon dioxide. And so chloroplast is another energy-related organelle. So in chloroplast, we're able to take sunlight energy and using sunlight, carbon dioxide, and water, plants and proteins and algae are able to convert that into glucose through a process known as photosynthesis. Now, this energy that it makes can then either be stored as starch, as we saw when we were looking at carbohydrates. It can be stored as starch, or we can actually move that energy into the ATP synthase and into these little vesicle called thylakoid. So the thylakoid membrane, similar to the mitochondria membrane, is double membrane and can actually do ATP synthesis there. And this is through a process called phosphorylation. So plants are also able to make ATP using glucose, but that glucose was created through sunlight energy, carbon dioxide, and water. Okay, so I have a question for you. What do you think will happen? Let me move my face out the way. What do you think will happen to a plant if you grow the plant in the dark? What do you guys think? So here I'm showing two different images. They're both sunflower seedling, and the first one is grown in the presence of light, whereas this one is grown in darkness, so without light. What do you guys think will happen between these two plants? Will the plant survive? Will it be able to produce energy? What do you think? So the seedling that's grown in the dark, remember seeds, we know that seed has a lot of calories to it and a lot of fat. Well, that seed, that originally has a lot of storage energy source, right? So originally the seedling is able to use that stored energy source in order to grow. So it's able to use that, but eventually that energy source will be depleted, right? And so the plant normally would do photosynthesis by converting, as we said, sunlight energy and carbon dioxide and water into glucose, right? Into more energy source. But without the sun, the plant will no longer be able to actually photosynthesize, or without any light, it won't be able to synthesize. So what you see is, you actually see, if you look at the phenotype between this guy and this guy, you actually see that this is elongating. Why do you guys think it's elongating like that? It's elongating because it's trying to find the sun. It's like stretching out, and it's like, where are you, sun? It's trying to find the sun that is not present because we're mean and we grew it up in the dark, right? So it doesn't have the sun, so therefore it cannot do photosynthesis. So I'm going to cross this out. Boo. No photosynthesis for it. And eventually, the chlorophyll that is normally present in plants that is able to do photosynthesis will kind of get depleted. And that's why a lot of times you see plants that are grown in the dark start to lose that green that we're used to seeing because it's getting depleted. So now these plants will actually lose that coloring. So the plant isn't happy, and eventually if you grow a plant in the dark, like if you stick your plant in the closet, it'll survive for a while because it has some energy reserve, but eventually it will die. Okay, so womp, womp, womp. We're going to move on to cytoskeleton. So these are protein structures that actually help the cells with its shape, as well as help with movement, not only externally helping the cells actually move along, but also internally helping moving organelles and DNA and stuff within the cells itself. So eukaryotic cells have a large network of interconnected protein filaments and tubules. And so there are three main different filaments that we're going to look at: microtubules, actin filaments, and intermediate filament. We'll break them down a little bit more in the next slide. But what this interconnected protein filament and tubule does is it allows the cell to maintain its shape, right, so that it doesn't collapse within each other. And it also allows the cell to move around within the tissues, as well as allows the cells to actually internally inside the cell move organelles around with different type of protein structure. So here we actually have three different pictures, and these are actually really neat to look at. So this is actually a cell, and the coloring is coming from a staining, so we can actually stain for particular organelles and tissues and proteins within the cell. So what you have here is this nucleus is being stained red here, and then all of this green is the staining of all the microtubule that's in the cell. So this whole thing is a cell, and these green stainings just showing you how much microtubule is actually in that cell, right? So again, we have it here. This time the nucleus is stained blue. This is the cell membrane that we're staining right here, that's red. But then all of this green, again, is showing you the different filaments, and it's being stained. Again, right here, we can see another one. So these are all just kind of showing you, what I hope you appreciate, just how much cytoskeletons we have there. Just all these different tubules and filaments and actin. So let's kind of look at these cytoskeleton filaments in a little more detail. So here I'm showing you three different ones: actin, microtubules, and intermediate filaments. So the microtubule is the largest one of them all, 20 nanometers in diameter, and it's the thickest one. It's the one that is going to help with regards to transporting water and ions and small molecule. Also, when we're doing cell division, when the cell is dividing, it is the filament or the tubule that's involved in actually kind of forming the ester that we'll actually see, and that will become the meiotic spindle that will actually come in and kind of grab onto that DNA and pull it into the two different cells, right? And then we also have it being the foundation or the basic structural unit for centrioles, which again has to do with division, as well as cilia and flagella. So we're going to look at cilia and flagella in the next slide. Next, we have the intermediate filament, which is this guy right here. This one is not as big as the microtubule, so it is 10 nanometers in diameter, and intermediate filament plays a role with regards to contributing to the cellular structure. So it actually plays a role in kind of holding tissues together, such as in skin. And the last filament that we're going to talk about is known as the microfilament. It is the thinnest one. It's 7 nanometer diameter. We actually see it here as actin filament, but it's the thinnest one, and it's the one that actually helps maintain the shape of the cell the most. So it kind of helps with the shape. It also helps contract the cells, mainly in muscle cells. And lastly, it also kind of helps with moving the cells around. So for example, when we have infection, the way that white blood cells are able to migrate to the site of infection and kind of help with the pathogen is through these microfilaments. So as we mentioned before, we're going to talk about cilia and flagella of microtubules. So cilia and flagella are found in the cells to help with cell movement. So we find it in a eukaryotic cell. Both of these are made out of microtubules, as we mentioned, and so we can see it here that we have cilia, which are shorter and they're a lot more abundant compared to flagella. Flagella is like nice and long, as we can see here. So we can find a lot of cilia within your lung tissue, like along your bronchioles, and we also can find a lot of cilia in different within your digestive system, to kind of help with regards to moving those molecules around there too. We can find flagella, for example, in sperms. So the way the sperm can kind of move throughout the body is it has a flagella at the end that kind of spins really fast, and that momentum is kind of what propels the flagella. So both of these are actually made of microtubule. And so this video here will kind of show both of them being moved in a protozoan. So it's actually really interesting to watch. So go ahead and watch that video on your own time. But you can kind of see here, this is a close-in of this flagellum, and you can see within the flagellum we have these little motor proteins, but we also have a lot of like microtubules in there, right? So we have like these microtubule pair as well as the central microtubule. So when you hear flagella or cilia, just kind of think of movement.
The flagellate protozoans, also known as...
Okay, so this network of proteins and polysaccharides, which are sugar, right? Remember our polymer of sugar or carbohydrates called polysaccharide. So this network of proteins and sugars is what allows the cells, as we mentioned, to have shape and structure, right? So collagen, for example, that we can observe here is kind of what provides stretching and allows these cells to stretch a little bit. And it also provides elastin, so it helps to provide so that cells, when they are being pressured by different outside force and fact, that they're able to keep that shape, and they're able to stretch, and they're able to kind of keep that resilient within their shape, right? Even under different type of pressure. And so a lot of times we can see collagen actually working by kind of pinching our skin and kind of seeing how the skin is able to bounce back. And unfortunately, as we get older, our cells does start having a harder time making these different proteins and polysaccharides. And last, we're going to kind of look at cell junction. While cell junction itself is not part of, not one of the filaments that we consider part of the cytoskeleton, these junctions what allows the cytoskeleton within different cells to actually work together to form a network. And so you have these different junctions. So when you think of junction, just think of how we are able to kind of have the cells attached to one another and communicate with each other. So the first one is adhesion junction. So this is actually going to hold the cells together by actually attaching. You can see here is actually attaching the cytoskeleton together. So we see that happening right here. And then you have tight junction, which is when you actually have the junction, we have these proteins that actually combine together. So it's almost like the zip tying the different cells. So this is one cell right here. This is the other cell over here. And this is almost like a zip tying these cells together. And the last type of junction we have is a gap junction. So this is the one that's actually found in your heart. So this gap junction, they're actually these junctions, but you can kind of see if you look at it closely that there seems to be a line in between. So this functions pretty much as a channel that allows the cells to actually pass back and forth different ions in order to communicate with one another. So this is found a lot in the heart because if you think about the heart has a lot of different cells. Well, how, like each time that the heart contracts and beats, how does it know to beat all in sync? Well, it's because we're able to actually communicate, the cells are actually able to communicate with each other through these gap junction. So we find this a lot in a lot of different smooth muscle and in heart muscle, having these junction with these different membrane channel that allows the ions to be passed back and forth as the way that the cells can communicate with one another. Okay, and this is the end of the section for the last section for chapter 4.